into the reproductive system. So this is a weird system. This is a system that actually doesn't really kick in until we hit puberty. So you hit a certain period of time where your brain says, okay, now it's okay to reproduce, right? Sound like your mom. All right, so we have gametes. Gametes are actually cells that only have 23 chromosomes, right? Because we're going to have a male gamete meet a female gamete so that we can have 46 chromosomes. That's compatible with life. The term, the anatomical term for sexual intercourse is copulation. Let's see. After a sperm meets an egg and there is fusion of both of their pronuclei, it's going to become a single cell called a zygote. And that doesn't last very long because very quickly, as you've maybe you've already watched the development video, very quickly that zygote is going to start going through mitosis, where that one cell where sperm and egg come together. Now we got 46 chromosomes. Very quickly it's going to start to go through cleavage, forming blastomeres. So one cell is going to become 2 cells, 2 cells is going to become 4 cells, 4 cells are going to become 8 cells, blah, 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 blah. And the term development of a fetus, so what we went through with development in lab, going from that zygote, going through all those differentiations, that's called gestation. And then giving birth to a baby is called parturition. Okay, sometimes we've mentioned homologous structures. When we talked about meiosis, we talked about homologous chromosomes. So a homologous chromosomes was essentially carrying the same information. Well, in embryology, what we're talking about is there is the same general tissues that will eventually develop into something different. But originally, our gonads came from the same embryonal tissue. So a male testicle came from the same embryonal tissue as a female ovary. Yeah, this also includes like the penis, and I mentioned this in the earlier reproduction lab, how the clitoris of a female, so the stimulatory organ, came from the same embryological structures as the male penis. It's the same stimulatory organ. Okay, so what happens inside those primary sex organs, the testicles and the ovaries? We're going to form the gametes, the things that have the cells that have 23 chromosomes that can come together to make a baby. And they produce hormones, and you already know this. Males make testosterone, females make estrogen and progesterone. Men got it easy. Men are very simple as far as, like, when you're studying anatomy and physiology, it's pretty cut and dry. Females were complicated. I'm going to start with the male reproductive system. It's just easier. We have other things besides our testicles and our ovaries. We have accessory. In our hypothalamus, we have that GnRH. You've already met him. He is in charge of those sex hormones, and he's that FSH, follicle stimulating hormone, is going to be in charge of making the ovum and making the sperm. So the GnRH tells the pituitary what to do. The anterior pituitary is going to release the follicle stimulating hormone and the luteinizing hormone. This is the part where I told you in the very first week of school when we were doing endocrine, I said change that interstitial cell hormone into LH, because we have discovered that LH is the same as interstitial hormone. Blah, blah. Okay, so when do our reproductive organs actually become functional at puberty. So females are pretty early, just the whenever the menstrual cycle kicks in. So as early as whatever, 10, 11 years old, as late as 18 years old, everybody's different. And then men, when you start to hear that voice crack, we're getting pretty close to that time. You already know this part. This is nice. Sister chromatids. During interphase, at the very end of interphase, DNA needs to replicate before we can start to go through divisions. Divisions in meiosis is a little bit different from mitosis because of the way that the sisters line up. So in mitosis, when your skin cells, you need to replace your skin cells. All 46 pairs, so 92 chromatids, but 46 chromosomes with the centromere in the middle, they all line up in the middle during metaphase. With meiosis, you're going to have 23 pairs on the left side of the equator, 23 pair on the right side of the equator. How do they line up? Well, they're going to line up based on homology. Am I a homologous chromosome? This is my mom's chromosome that has the I allele. This is my dad's chromosome that has the I allele. They look similar, the same in length and thickness and everything. They'll line up, they find their homologous pair, moms to dads. 23 are going to line up in the middle. So what's the difference in this, mitosis versus meiosis? Meiosis goes through everything twice. So we've got, instead of having 46 in the middle, we're going to have 23 on either side. And when this, in prophase, when those 23 sets line up, so your homologous pairs, where each chromosome has the basic same genetic information, say, just making this stuff up, but let's say homologous number 1 has, from mom, has the shape of your pinky toe and the shape of your earlobe and the color of your eyes and the color of your hairs. And the same thing with your dad. Those two guys are going to meet up in the middle. Now remember, they've doubled, so they have sisters and they're connected by a centromere. So we're going to have 44 little sticks together, but 2 of them are going to be binded by a centromere. So you've doubled your DNA. So essentially it looks like an X. 1 X on the left side, 1 X on the right side. So homologous pairs, 23 on either side during prophase. And then what's going to happen during prophase, they're going to exchange information. Here's my phone number, here's phone number, whatever. So what this is doing is creating diversity so that all of your children don't look like little clones of you. Now, clearly, some people weren't very good at it because you can look at some families and go, oh, wow, each and every one of those girls look like mom. Each and every one of those boys look like dad. Or sadder, each one of those girls look like dad. But anyway, whatever. Okay, so that happens during prophase I. After you're finished with prophase I, then you're going to have to separate the sisters now. And if you remember from mitosis, when you separate the sisters, the chromatids, actually become their own chromosomes once they're separated. Okay. Here's a picture of people with 46 chromosomes. Don't have a pointer, but if you can look at number 1, there's 2 sticks. One of those came from mom, one of those came from dad. Number 2. One of those came from mom, one of those came from dad. But during prophase, there were some little changes that happened that made them not quite like mom's, not quite like dad's. Chromosome after 22 where it says X. So this is set number 23. One came from mom, one came from dad. In this case, dad gave his X allele. The one that says that my baby's going to be a girl. 2 straight sticks. At the bottom here, chromosome number 23 that has the arrow coming off of it, dad didn't give his X allele. He gave the Y allele. See how short it is? But this is the sex chromosome. So on number 23, where we determine, is this mom or is this dad? Or, sorry, is this a boy or is this a girl? Sorry. If you have 2 same size sticks, then that's going to be a girl. But the male is a lot smaller. So on this homologous chromosome, this sex cell chromosome, this chromatid, is going to be a Y chromosome. So this baby is a boy. So if you have a boy, then your mapping, your genomic mapping, looks like this. Okay, you'll see the term haploid cell. Well, a haploid cell is the kind of cell that only carries 23 chromosomes. A haploid cell is only a ovum or sperm. A diploid cell, so 2 di, is going to be 2 times 23, 46. So most of our cells are diploid cells. Now this is talking about how a zygote is diploid. Well, of course it is, because we've taken 23 from mom, 23 from dad, we put it together, we made a 46 chromosome cell. All of your cells are diploid, except ovum and sperm. Now, if you've watched the Amoeba Sisters video, you should be good at this now. The most complicated thing that I feel like they don't explain extremely well is that mitosis, all 46 chromosomes line up at metaphase. In meiosis, 23 line up from mom, 23 line up from dad. They've multiplied, so they have sisters, and they'll cross information, and they do fine in the prophase part. They switch information, but all 46 don't line up in the middle with meiosis. With meiosis during prophase, they find each other. Hey, I'm like you, you're like me, let's be buddies, let's exchange information. Whoa, we got a line up in the middle, that's metaphase. So during prophase, they exchange that information. During metaphase, they're going to line up in the middle in pairs, like they're going to do a dance. Then everything is pretty much the same after that. So this is just a picture showing how in interphase, they doubled their information. So now instead of looking like sticks, like chromosome sticks, now they look like X's, but only because they've doubled and they're still stuck together by a centromere. Remembering from AMP1, if there is a centromere, that counts as 1 chromosome. After meiosis 1 is done, then you've separated the pairs that were paired up during metaphase, but you still have to separate the sisters. So now with sisters, you're going to separate the 2, and now you have 23 chromosomes per haploid cell.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase I, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Like mitosis, meiosis is preceded by an interphase, during which the chromosomes replicate. The centrosome also duplicates in preparation for cell division. Prophase I begins with condensation of the chromosomes. Homologous chromosomes, each made up of two sister chromatids, come together in pairs. This pairing is called synapsis. Each chromosome pair is called a tetrad, a complex of 4 chromatids. Chromatids of homologous chromosomes cross over each other and exchange parts. Meanwhile, other cellular components prepare for the division of the nucleus. The centrosomes move away from each other and spindle microtubules form between them. The nuclear envelope and nucleoli disperse. Finally, spindle microtubules capture the chromosomes, and the chromosomes begin moving to an imaginary plane equidistant from the poles. At metaphase I, spindle microtubules from one pole of the cell are attached to one homologous chromosome, while spindle microtubules from the other pole of the cell are attached to the other chromosome of the pair. Thus, the homologous chromosomes are poised to move to opposite poles of the cell. During anaphase I, each pair of chromosomes is pulled apart, and the homologous chromosomes move toward opposite poles. Note that sister chromatids remain attached at their centromeres and move as a unit toward the same pole. The chromosomes finish their journey during telophase I, and cytokinesis occurs, producing 2 haploid daughter cells. Note that each chromosome still consists of 2 sister chromatids. Meiosis isn't over yet. Remember that it consists of 2 consecutive divisions. During meiosis II, the sister chromatids will be separated. The second division of meiosis, meiosis II, is essentially the same as mitosis. The important thing to remember is that meiosis II begins with a haploid cell. During prophase II, a spindle forms in each cell and the chromosomes move toward the middle of each cell. During metaphase II, the chromosomes align in the middle of each cell. Sister chromatids are pulled apart in anaphase II. In telophase II, nuclei form at opposite poles of each dividing cell, and cytokinesis splits the cells apart. Meiosis has produced 4 haploid cells, each with one set of chromosomes.
Yeah, you got through a bunch of slides there, because I can't play those, but you can. All right, so this is just repetition. In meiosis I, the goal is to make 46 chromosomes become 23 chromosomes because they're going to line up in pairs, 23 pairs on the left, 23 pairs on the right. But while they're finding each other during prophase, they're going to share information before they have to do that lineup. So this would kind of be like olden days when you have to line up and do a dance like in Europe. You probably haven't watched any of those historical shows, and I'm dating myself. But before the actual dance happens, you might mingle with that cute boy or that cute girl and exchange some information. Prophase. Now it's time for the dance. So now we have to line up side by side along the equator and do all the formalities. That would be metaphase. All right. So you've got 2 for mom lining up on, or you've got the sisters lining up for mom on the left, dad, sisters lining up on the right. During metaphase, anaphase, they all get pulled apart. So at the end of meiosis, each daughter cell has chromatids, so that's a chromosome, but we have separated 23 on the left, 23 on the right. That 23 is not 46, so that makes it N. N is 23, so we're haploid already. We still have to separate the sisters, though. So then we're going to go through meiosis II. When we go through meiosis II, then we separate the sisters. Here we go, separating the sisters. And now we have 4 daughter cells that each have 23 chromosomes. So mitosis is used for almost all body cells except the sex cells. Mitosis is about repair and replacement. You know, your skin cells, they die once a month. They're gone, done, poof, gone. You got to make some new ones. So that's about mitosis. Or maybe you have a liver cell that's just worked himself to death because you've been, whatever, just taking too many medicines, or working with too many chemicals, or drinking too much, whatever. So you've killed those liver cells, you gotta make some more. Meiosis is only about sex cells. Also with meiosis, we have to remember we're creating genetic diversity during prophase I. We're doing the crossing over, and we're making sure that each of those final daughter cells are a little bit different. Okay, let's get into the specifics of a male. So a male has different things. The testicles are producing the sperm cells. So they're making the final haploid type cell that's going to meet the female's haploid type cell. But they got to get delivered somehow. So a couple of things that I'm going to talk about. Epididymis, you've probably never heard of that guy before. The ductus deferens, you've heard of him before, but you've never really thought very much of him. This is what when men get their vasectomy, this is another name for a ductus deferens, is a vas deferens. So what they do is they tie off the vas deferens during a vasectomy. Okay, ejaculatory duct, you haven't really thought about him very much, but what he is combining fluids with the sperm to allow delivery of the sperm. So So it's full of volume. And of course the urethra, you've already met that guy. Now you haven't, unless you listen to the I Hope You Have Reproductive Lab, then you haven't heard of these guys. But we have seminal vesicles or seminal glands. We have the prostate, and they have the bulbourethral glands, which in your lab book is called Cowper's glands. I like bulbourethral glands, probably because that's how I learned it. But I never heard the Cowper's gland until I started teaching this class. I was like, what the heck is that? Oh, bulbourethra, I got it. Okay. All right. So here is the different glands. Now, in the lab models, I told you that you weren't going to be able to see the Cowper's glands. Look at him. That's why he's little, and he's right at the base of all the penile tissue, and there's one on the left and one on the right. So these models that we have in lab, they're mid-sagittal cuts. So since they're mid-sagittal cuts, we kind of miss that guy. Okay. So the scrotum is actually the sac that the testicles sit in. And they have actually a very important job as far as reproduction goes. He has a muscle embedded in him that can contract and relax. There's actually two, but there's one that's actually part of the scrotum itself, and there's another one that actually raises up kind of more superiorly. Both of them are in charge of making sure that the temperature for those testicles is correct for reproduction. One of them is called the dartos muscle, and the other one is called the cremaster muscle. So if everything is too cold, so you just jumped into an ice cold river, those muscles are going to contract and bring the testicles closer to the body to protect them, because they need warmth to produce sperm. However, if it's really, really hot, if things get too hot, then they can't produce sperm. So most of the time the testicles hang down a little bit so that they're not hugged up right next to the body at 98.6. Bicyclers that wear tight shorts, they're on a tight seat, and their testicles are too close to their body, then they're going to have potential problems with reproduction because their testicles are staying too hot, and so they cannot make sperm. Okay, so this is a setup. Wish I had a pointer, but I don't. But you can read. You can actually see the cremaster muscle and the dartos muscle. The dartos muscle is much more superficial and kind of inferior, and the actual cremaster muscle goes all the way up through the inguinal ring. So, for example, when I'm doing a neuter, a dog neuter, I can actually visualize that cremaster muscle, and I'm actually pulling him away from the testicle. I don't mess with the dartos muscle at all because it's really superficial. It's part of the scrotum. When I do a neuter, I'm going internal. So I can relate to this. They don't neuter men very often unless they have some sort of medical problem. And even then, they go into the abdomen. They don't mess with scrotum. So wish I had a picture, but I don't. I see the cremaster muscle all the time. I have to separate the cremaster muscle off of the testicle, and then I neuter the dog. I don't mess with a dog's scrotum. If you cut a dog's scrotum, when they wake up, they're going to chew themselves open because you've touched their scrotum. It's a very sensitive area. You don't mess with the scrotum. Don't touch the scrotum. Okay, this one's probably got a little bit too much information as far as the tunica vaginalis and the tunica albuginea. If you're going to neuter somebody, then you need to know these things. But let's move forward to when you cut into a testicle, you have all these tiny little separations that you could see well with a microscope. But on your lab 2 microscopic slides, you can see all those seminiferous tubules. This is where sperm is being made. So I've cut into the testicle, and you have all these little tiny little separations where you have seminiferous tubules. This is where the spermatogonia are that are so busy all through a male's life up until their 70s, maybe even 80s. Females' oogonium don't work anymore past embryonic stage. They're done. What you're born with is what you got. Oogonium, finished. Spermatogonium, very busy. So, we got people like Mick Jagger making babies in their 70s. Okay. Well, after you make a spermatid, he still has to learn how to swim. So he's going to go out of the seminiferous tubules to this little storage area called the epididymis. And here is where they're going to learn how to swim and actually be stored until ejaculation. Spermatic cord is really I'll show you a picture of it in a second, but this is an area that's part of the vas deferens. You can separate it out from the vas deferens, but it's this truly like it looks like a cord. It has the cremaster muscle in it, and it has lymphatics in it, it has artery, veins, and nerves. It has something called piniform plexus in it. I think I just moved the slide. But anyway, this would be important if you were learning how to neuter somebody, but I'm going to skip it for this lecture test. Okay, so here we are, seminiferous tubules. This is what you see when you look at the microscopic slides for lab exam 2. You should see a bunch of these seminiferous tubules. Okay, the actual circle part is the tube. You cut through it. I've already mentioned the spermatogonia in the lab manual. The spermatogonia are stem cells. They're going to go through mitosis. One guy gets to stay a stem cell. The other guy is going to work its way into a primary spermatocyte to eventually become a sperm. The term genic is the birth of, just like everything that you learned in A&P 1. Okay. So you're looking inside the seminiferous tubule. Inside the tube, but at the periphery of the tube is where you're going to find those spermatogenic cells. There's little cells that are like helping take care of the spermatogenic cells. They're called nerve cells or Sertoli cells, as I have it bolded. So just like, do you remember A&P 1 where we had neurons? And in the central nervous system, we had astrocytes that were taking care of the neurons. In the peripheral nervous system, we had satellite cells that were taking care of the neurons. What do you need? Do you need some water? Do you need some glucose? Do you need some sodium? Whatever. Taking care of the cell that needs to do the very important job. Nervous system, neuron, needs to fire some sort of electrical impulse. He can't do that if he doesn't have all the nourishment he needs. Same thing with the spermatogenic cells, spermatogonia. If he doesn't have everything he needs, he can't make a sperm cell. How good is that? Then we're all going to die. Okay, so we're going to have these Sertoli cells making sure that we're taking care of that. The Sertoli cells are going to respond to follicle-stimulating hormone, which help with the formation of sperm. I have everything in little right here because your book goes into a lot of details that is not going to show up on your lecture test. But in essence, he's going to make this protein that makes testosterone, not estrogen, testosterone in high concentrations. You need testosterone in high concentrations to make the sperm cells. Meiosis cells, forget about those. Okay, so then we have these tubes, okay? We've got these seminiferous tubules. in little circles. Well, of course, there's cells between the circles. Outside of the circles, we have these other cells that are called Leydig cells. They used to be, well, I guess not used to be, it depends on which book you got this semester. Sometimes they're called Leydig cells, sometimes they're called interstitial cells. These guys are producing testosterone. We would not be able to do a good job of keeping our manliness without testosterone. And they respond to luteinizing hormone. If you remember at the beginning of the semester, we had the interstitial cell stimulating hormone, and I said scratch that out, make it LH, because we found out that interstitial cell stimulating hormone is the same thing as LH, luteinizing hormone. So we don't need two different names. And this is nice because Leydig cells, the person that discovered all this stuff, LH, LL, luteinizing hormone, it all matches up. And then, okay, here's a picture. You can read it yourself. All right. Same picture, but this is in reality. This is actually a cadaver. So notice how the epididymis is like an extension coming up. It almost caps the top of the testicle right there. And this is that sperm school area that I was telling you about. It is extremely evident. So people don't get neutered very often, but dogs do. They should. And you can definitely see an epididymis. Same thing with a horse. It's very large. It looks very different. This is where sperm learn how to swim. This is how they actually get their motility. And then also this is where they're stored until ejaculation. And then, so this is in your the thing in lab that says microscopic slides for lab test number 2. This is a seminiferous tubule. So on the most outside part are the stem cells that are going to make sperm. They've got to go through some changes. In between each little circle, we've got other circles that are Leydig cells. So they're going to secrete testosterone and do the manliness and whatever. So I say manliness. I'm minimizing it, but I shouldn't. So men have chest hair. Thank goodness I don't. Whatever. Chest or hair on the back. The deep voice, the changes to the larynx, whatever that makes a man a man. Myoid cells, don't worry about that. Is myo. Myo is muscle. So you can figure that out. Myoid like a muscle. Okay, so initially, spermatogenesis is the development of a sperm cell, and that's happening in the seminiferous tubules. I bet you've already figured that out by now. Spermatogonium go through mitosis. One guy stays a stem cell, the other guy is going to become a primary spermatocyte. Then we're going to start meiosis. We're going to go through meiosis 1, it's going to go through meiosis 2, make 4 spermatids. This happens throughout a man's life. Okay? We don't have tails yet. We are going to develop those tails later, and that's true spermiogenesis. So not spermatogenesis, the spermiogenesis when you get the tails. Splitting hairs, who cares? Not on your test. So spermatid are those 4 things that are made from meiosis, but they're not fully developed. We have to finish developing this guy. We need a tail. Now here is test material. You're going to have the head of the sperm, which is where the DNA is. We're going to have a cap on the sperm. It's called an acrosome. And why is he important? You can't get through the egg's zona pellucida without it. You need the acrosome because he has digesting enzymes that can actually break up the zona pellucida so he can get his 23 chromosomes in with her 23 chromosomes. The mid piece, the middle of that sperm, That's where the mitochondria is. That's where all the energy is. And then the tail is the guy that helps the guy swim. You don't have a tail, you ain't getting there. You got a long way to go. You got to swim all the way up the vagina. You got to get through the cervix, which sometimes has still some mucoid plug. Then you got to get up into the oviduct / uterine tube / oviduct, uterine tube, blank. Anyway, you're probably tired of hearing me saying it anyway. You've got to go through all of that up towards the top third of the oviduct. So you got a long way to go. If you don't have a tail, it's not getting there. So here's showing the development of the sperm. Once you're done with spermatogenesis, all you have is spermatid. You've got to go hang out in the epididymis to learn how to swim. You're going to develop a little tail, and you're going to differentiate into a true sperm, a true swimming sperm. And then, overall, from the very beginning, going through mitosis from that one spermatogonium, dividing into a spermatogonium and a primary spermatocyte, all the way to the swimmer, is approximately 2 months, maybe a little bit more than that. Now we're going to have some fluids that are going to help this guy travel too. Here's great things for fill in the blank. Testicular cancer. This is rare, but honestly, usually when it happens, it's going to be a pretty young man. Things that can make you prone to testicular cancer, sometimes it's just bad luck. But if you got mumps, be current on your vaccination. If you get mumps, you can potentially get an infection in your testicle. Infection or inflammation. And that's going to create swelling, tenderness of the testicle. And that any inflammation, we know that inflammation can eventually lead to cancer. Inflammation of anything, inflammation of skin, sunburn, can lead to skin cancer, just for example. Inflammation of the uterus can lead to uterine cancer. So anywhere you have inflammation on a pretty routine basis, that makes that tissue more prone to cancer. Something significant like the mumps, that's a pretty bad virus. That can lead to orchitis, which is inflammation of the testicle, and that can lead to cancer. Cryptorchidism, look, it's black, so it sure could show up as a fill-in-the-blank question. Super common. To be cryptorchid, it means that one of your testicles didn't come down. During embryonic development, coming from homologous tissues, testicles and ovaries are the same tissue. Do the ovaries descend down into a little sac outside of the body? No. Homologous tissue. The female's gonads stay inside the body. There has to be more changes with the development of a little boy, and the testicles are supposed to go down this little tract through the inguinal ring and then into the scrotum. Sometimes that doesn't happen. And so one of the testicles is going to stay in the abdomen, or both. But anyway, when testicles don't come into the scrotum, whether it's one or both, more commonly just one, that's called cryptorchidism. And so when the testicle stays inside of the abdomen, that testicle is not in its appropriate environment. And so it is more likely to actually become cancerous. Now, when a baby is young, they can actually try and get this testicle down into the scrotum. But if a parent doesn't, got to take your kid to the doctor. Parent doesn't take their kid to the doctor, and they're like, well, whatever, he's different. That testicle sure could become cancerous. Infertility. There's so many different reasons that a couple can be infertile. It could be mom, but it could be dad. It doesn't matter. For different reasons. Age is a factor in a male, but you know what? Men can still make babies in their 70s. So women are often the other problem. My daughter's was taking a class at A&M, and I hate to scare y'all, but one thing that they've discovered is the residue from receipts actually has a toxin in it that can make a man infertile. If that isn't scary. Now, I'm sure it's rare. Maybe you'd have to eat 1,000 receipts. Maybe you have a pica problem and you like to eat receipts. I don't know. But anyway, you have lots of different reasons, lots of different chemicals in the world, and overexposure to any of these chemicals could cause a problem in males or females. Other things that are pretty common, if you know you're going to go through radiation or chemotherapy because you have cancer, then they're probably going to have you bank sperm for a later date, just in case you are rendered sterile from the treatments, from the chemotherapy or the radiation therapy. Then you can do in vitro fertilization from your own personal sperm that's been frozen. You know, if you marry and then, you know, you want to have a baby, it's still your baby. Nothing, absolutely nothing wrong with adopting. But if you want your own baby, you're going to have to bank, because if you're going through radiation or chemotherapy, then you're going to have to save that. Excess alcohol, marijuana, any chemical can mess with the sperm. So your sperm could end up with, all your sperm could end up with multiple tails, multiple heads. They don't know how to swim, whatever. You already have some boo-boos where some of the sperm occasionally come out with 2 heads or 2 tails. And clearly there's going to be directional problems. MAP won't help to get the sperm up into the oviduct. Other things, like I said earlier, if your testicles stay too hot all the time, then you're going to have a problem with making good sperm. And then weird things like having problems with calcium channels. So we mentioned calcium being important for blood clotting and being important for firing a nerve, making a muscle contract, but we hadn't mentioned it at all, and I'm still not going to go past this for sperm, but you need calcium for sperm production. Okay, so the duct system. Epididymis. So we've made the sperm, they've been hanging out in the epididymis, they've learned how to swim, they're ready. Okay, with ejaculation, they're going to travel from the epididymis through the ductus deferens, through an ejaculatory duct, which is going to come out of the prostate into the urethra. The ejaculatory duct is the link from the reproductive tract to the urinary tract. The ductus deferens, also known as the vas deferens, is what is going to be tied off during a vasectomy. Okay, the urethra. You've already met the urethra, but you didn't know they had a bunch of names. There's this urethra that is going to be inside the donut of the prostate. So the prostate is immediately inferior to the bladder. It's a little donut around the urethra. That's the prostatic urethra. Momentarily, it's going to be called the membranous urethra. I like that name better than the intermediate part of the urethra. That's too many words. Membranous urethra. And then as it's going through the penis, you could call it the penile urethra. It's also called the spongy urethra. Picture, you can read it yourself, but notice, I don't have a pointer, but notice the little donut of a prostate that is immediately inferior to the bladder. Notice the seminal vesicles. It says seminal gland, but seminal vesicles. Notice the bulbourethral glands. And for a moment, I need to pause. Lab. So the bulbourethral glands, also known as the Cowper glands, are important for lubrication. Initially with stimulation, these are the first guys that are going to release some fluid. It's kind of a mucoid fluid for lubrication for penetration. Then we're going to have the prostate is going to release some fluid that's going to help change the pH of that semen so that all the sperm don't die when they hit the vagina. The vagina is very acidic. So the prostate is going to help, the prostatic fluid is going to help buffer that semen to make the acidity of the vagina less acidic and tolerable for all those little guys swimming inside the fluid, the semen. And then the seminal vesicles are labeled here as the seminal glands. Their job is to offer some fructose as nourishment to the sperm. So all three of these guys are going to offer a fluid which is going to make semen, and semen is what's delivered into the vagina, and it's going to give a nice healthy environment for these sperm so they don't die once they hit the vagina. I got some nutrients, I got a change in pH, and well, I guess the Cowper's gland is more for the female, whatever. This. Okay, so external genitalia is penis and scrotum. Men have more external genitalia than females. Females, everything's on the inside for the most part. The very tip of the penis is called the glans penis. It's usually covered up by something called the prepuce when a baby is born. Another name for the prepuce is the foreskin. But in America, most of the time that gets cut away because we've learned that the prepuce, this extra piece of skin, is going to harbor bacteria and dirt, and it's going to lend itself to more urinary tract infections. So it lends itself to uncleanliness, which infection. What started off as a ritual by the Jewish tradition, circumcision, now most people in America recognize for health reasons, let's go ahead and do a circumcision, remove the prepuce for health risks. Not just HIV. This slide says HIV because that's what's in the book. But from someone that used to babysit a child that was not circumcised, that boy always got urinary tract infections. Urinary tract infections in general are more of a girl thing because our urethra is so close to our anus, and so we females are more prone to bacteria getting into the urethra and moving up to the bladder. Boys, that their urethra is far from their penis, it's not a big deal. But if they're not circumcised, that prepuce is going to hold on to bacteria and dirt. Where's it going to go? It's going to travel up into the urethra, up to the bladder, urinary tract infections. So this little boy that I used to babysit, constant bladder infections, he got circumcised, life is good. Why was he not circumcised? They weren't from America. They were from somewhere else. Okay, so around the urethra, we have some tissues that are full of spaces. We have one that is around the urethra all the way, and that's called corpus spongiosum. And then we have one that is, it's a pair also of tissue called the corpus cavernosa. These areas are going to fill with blood, especially the corpus cavernosa during an erection. In corpus cavernosa, spongiosum, their job is to protect the urethra. Seminal glands, pretty big on a rat. Please look at your lab book. This guy is in charge of producing fructose to make sure that those sperm have all that they need. 60 to 70% of the volume of semen comes from the seminal glands. The prostate is this gland that is encircling the urethra immediately inferior to the bladder. And as the lab book notes, he's going to raise the pH to protect the sperm from the acidic vagina. Bulbourethral glands, all about lubrication. That's their biggest play. Okay, so here's that picture again. And the rat, again, that seminal gland is really big. On the rat also, you can see the prostate more inferior to the bladder. It looks a little different. Be sure and look at the lab book, because I truly don't know what's going to be on that lab test, but I do know the proper thing to say. If it's in the lab book, it's fair game for the test. Okay, benign prostatic hyperplasia. That means that your prostate gets bigger and thicker as men get older. So it's about half the men that are over 50 have this, and they need to go pee-pee more during the night. Because what happens as he gets bigger, when men go to the bathroom to urinate, they don't empty their bladder all the way. So if your bladder's not empty all the way when you go potty during the day, it's going to fill up quicker at night because you never emptied it to begin with. Prostatitis is truly inflammation of the prostate. Different reasons. Prostate cancer is the third most common of cancer death in men. It's a really slow cancer. So it's slow to grow, but the problem with cancers that are slow to grow, they're hard to kill. Fast-growing cancers actually respond to chemotherapy or radiation therapy faster than really slow-growing cancers. Digital exam is the best way to tell if there's something growing in the cancer. In the prostate, because you can feel differences. PSA levels have historically been the way to check, but sometimes you can have a high PSA level and actually nothing ends up being wrong. It's a great screening test, but in the end, it's not perfect. Okay, the term semen is actually the sperm and all the fluid that comes with it. So another name for it is ejaculate. So with ejaculation comes out semen. Loaded with sperm, normally. Usually approximately 4 to 5 mils. It could be less, but look at that number of sperm. So surely one of those 150 million guys are going to figure out how to get where they're supposed to get without asking for directions. I've already said the rest of this. We need some fructose to help things out. We need to change the acidity so that the sperm cannot be killed by the vagina. So we're going to make that fluid more alkaline. Great. An erection happens with so men are visual creatures, definitely. Their brain is stimulated by parasympathetic neurons. It's crazy, driven like by nitrous oxide, just like a fast car. See, why do men like cars? Okay. So what's going to happen? Erection happens with parasympathetic stimulation. And we're going to have this, the dilation of our arteries in the penis. When those guys, you've probably heard me say, arteries, veins, nerve all run together. Well, arterial dilation makes the arteries wider in diameter, and they actually end up pinching off the veins that are right next to them. If veins can't drain, just like when you put a rubber band around your finger, what happens to your finger? Your finger gets stiffer. So this is the same thing with this parasympathetic stimulation. The arterioles dilate, which they actually physically pinching off the veins that are next to them, and so blood kind of pools in the corpus cavernosum, and makes everything stiff. Okay, the term ejaculation is simply orgasm. So when the semen leaves the male's body. This is a sympathetic response. I'm going to tell you something. I had an anatomy professor in veterinary school, and he's a very unique man. And without going any further than that, he taught us point and shoot. P, parasympathetic, is going to be an erection, point. Shoot, sympathetic, is the true ejaculation, orgasm. Won't take it any further than that. So why do we not get urine in the ejaculate? Why does that not happen? Well, we actually have this internal smooth muscle that's going to constrict. It's in the bladder. You're not in charge of it. It's going to tighten down, preventing any urine from entering into the urethra during ejaculation. So it's a sympathetic response. We're going to tighten the smooth muscle at this sphincter. It's a smooth muscle sphincter. You're not in charge of it. And then here's the thing. After men ejaculate, they have this period of resolution where they absolutely cannot have another orgasm. Every man is different. It can last minutes to hours. That's called the latent period, this refractory period, this latent period. And that latent period extends with age. So the ability to ejaculate, so for a man to have another orgasm becomes longer and longer as time passes on in age. Erectile dysfunction. Now I'm going to tell you there's lots of factors that go along with this. So alcohol and drugs, different kind of drugs. So things that certain, not all of, but certain blood pressure medicines, so antihypertensives, certain blood pressure medicines, certain drugs for depression, all these things can cause erectile dysfunction. But in a nutshell, what's happening is that nitrous oxide that makes a car go fast, that makes a boy go fast, they don't have enough of it either. So they can't have an erection. Now, sometimes chronically what happens is blood vessels get blocked. So just like atherosclerosis, you can get clogged arteries that lead to a heart attack. Well, your coronary arteries aren't the only things that are getting blocked. So what can happen is the vessels that are, the blood vessels in the penis are getting blocked. So if you don't have blood engorgement, you don't have vasodilation with blood engorgement, then you can't have an erection. So if it's a long-term problem that doesn't seem to be resolving, there are things that you can do for this. Anyway, chronic problems need to be addressed by a doctor. Diabetes, as always, can cause so many different things. And so this is one of them, erectile dysfunction. Viagra, how does Viagra work? Well, it increases the amount of nitrous oxide. More nitrous oxide, more likely to have an erection. Okay, and then finally, so effects of testosterone. Overall, so a young man going into puberty, some things are obvious. The secondary sex characteristics. They're going to start to get pubic hair, they're going to start to get facial hair, armpit hair, maybe even some chest hair. As time goes on, more chest hair. Their voice changes because the larynx actually starts to enlarge. Their skin gets thicker and oilier. Boys go through this amazing growth spurt at the end of high school, early college age. Men, I'm sorry gals, definitely have more muscle mass. It's just the way it is. That's testosterone base. And alas, they could eat salad for a week and drop 15 pounds. Women could eat salad for a week and drop 2. That has to do with testosterone. Their metabolic rate is just higher than females. And then, of course, young men definitely have because of testosterone, a higher libido. They're more of a sex drive. And that's it for the male repro tract. Watch, which is on the separate PowerPoint. You probably have this in well, you do. In mastering, you have this. You can watch it.